Yeah, welcome back, everybody. We're on to a continuation of Chapter 4. We continue with the topic, dideoxynucleotide procedure for sequencing DNA. A dideoxynucleotide is a human-made molecule that lacks a hydroxyl group at both the 2' and 3' carbons of the sugar moiety. In perspective of this, we have a figure here, 4.2, which shows a dideoxynucleotide. The 2' and 3' carbons of the sugar moiety lack hydroxyl groups, and a deoxyribonucleotide. And we have, in contrast, a natural deoxyribonucleotide has a 3' hydroxyl group on the sugar unit. Normally, during DNA replication, an incoming natural nucleoside triphosphate is linked by its 5' alpha phosphate group to the 3' hydroxyl group of the last nucleotide of the growing chain. However, if a dideoxynucleotide is incorporated and if a dideoxynucleotide is incorporated at the end of the growing chain, DNA synthesis stops because that phosphodiester bond cannot be formed with the next incoming nucleotide. The termination of DNA synthesis is the quintessential feature of the dideoxynucleotide DNA sequencing method, although other experimental conditions must be met before a DNA sequence can be determined. In principle, the first step in the standard laboratory procedure for dideoxynucleotide DNA sequencing entails annealing a synthetic oligonucleotide 17 to 24 mer primer to a predetermined segment of a strand of the DNA to be sequenced, for example, to a segment of a cloning vector near the insertion site of the cloned DNA. The oligonucleotide acts as a primer by providing a 3' hydroxyl group for the initiation of DNA synthesis. In the original method, the prime DNA is partitioned into, and before we can proceed to that, let's just finish off figure 4.21, which talks about normal DNA synthesis. An incoming deoxyribonucleotide or nucleoside phosphate ddNTP base pairs with the complementary nucleoside of the template strand. The internucleotide linkage occurs between the 3' hydroxyl group of the last nucleotide of the growing strand and the alpha phosphate group of the incoming nucleotide. Now, as we see, the prime DNA sample is partitioned into, and going on to the continuation of that, into 4 separate reaction tubes. Each tube contains 4 deoxyribonucleotides, the ADP, the CDP, the GDP, and the TTP, one of which is radio-labeled, and one of the 4 deoxynucleotides, so that is dideoxyadenosine triphosphate, ATP, and ddCTP, and the ddGTP or ddTTP. The concentration of each dideoxynucleotide in each reaction tube is carefully adjusted to ensure that it is incorporated into the mixture of growing chains at every possible site. Recall that chain growth stops as soon as dideoxynucleotide is incorporated, so each growing chain will eventually contain a dideoxynucleotide at its 3' terminus. This experimental condition is met in each of the 4 reaction tubes. Consequently, after each enzymatic DNA synthesis with DNA polymerase, each reaction tube will contain a unique set of single-stranded DNA molecules of all possible lengths, each of which includes the primer sequence at its 5' end. The synthesis reactions are stopped by the addition of formamide, which prevents DNA strands from base pairing, and the DNA molecules, including the newly synthesized DNA molecules, are separated by a polyacrylamide gel for electrophoresis. This separation procedure resolves pieces of DNA that differ in size by as little as single nucleotide. An autoradiograph of the gel shows only radio-labeled DNA fragments that were produced during the enzymatic DNA synthesis step. Each of the 4 lanes in the gel and the autoradiograph corresponds to a reaction tube that contains one of the 4 dideoxynucleotides. The sequence of a segment of DNA is determined by noting the order of the bands as accurately as possible from the bottom to the top of the autoradiograph. In the example shown in figure 4.24, the first 6 bases of the sequenced DNA are ATC, TGC. In this case, the fastest migrating band, the radio-labeled fragment closest to the bottom, which corresponds to the smallest DNA fragment, is in the ddATP lane. And the next band is in the ddGTP lane. The next is in the ddCTP lane. The next is in the ddTTP lane, and so on. Up to 500 bands can be resolved reliably on most autoradiographs. Usually the primer sequence is positioned about 10 to 20 nucleotides away from the target DNA so that the researcher can recognize the known sequence at the start of the autoradiograph and thereby identify precisely the first nucleotide of the target DNA. Although the procedure described above is still used for some specialized applications, in practice the entire procedure has been automated and it typically uses nucleotides labeled with fluorescent dyes that are detected using a laser rather than a radio-labeled nucleotide, which are visualized. And in an autoradiograph, automated DNA sequencing minimizes manual manipulations and increases the rate of acquiring sequence data, which is essential for assembling vast amounts of nucleotide sequence data. From whole prokaryotic and eukaryotic genomes. Automated sequence analysis can be carried out with 4 different fluorescent dyes, one for each dideoxynucleotide reaction, or with the same fluorescent dye for each dideoxynucleotide in each reaction mixture. In some cases, the primer rather than dideoxynucleotide is labeled with a fluorescent dye with a 4 fluorescent dye system. The samples of the collection of each reaction are pooled. In primer extension during DNA synthesis in the presence of dideoxynucleotides, each of the 4 reactions tubes contains a unique set of nucleotide extensions attached to the primer, because when a dideoxynucleotide is incorporated into the growing strand, it terminates the synthesis. A few full-length DNA molecules will be synthesized in each reaction tube, ddNTP dideoxyribonucleoside diphosphate. And this is the contents of each tube inside the primer and extension, primer and sequence of extraction. The figure 4.24 is that of a simulated autoradiograph of a dideoxynucleotide DNA sequencing gel. Each one of the gel was loaded with contents of one of the 4 reaction tubes, which contained either ddATP or CTP or GTP or TTP. By convention, the bands of the autoradiograph are read from the bottom to the top. In this example, the results of the sequence determination are shown on the right. And the fragments are separated in a single lane of a polyacrylamide gel or polymer-filled capillary tube. This type of analysis is called 4-color one-lane detection. Alternatively, with 1 fluorescent dye market, each sample is run in a separate lane. Right, going ahead with a milestone note is the specific enzymatic amplification of DNA in vitro, the polymerase chain reaction, courtesy of K.B. Muller's, F. Luna, S.J. Sharf, R.K. Saiki, G.D. Horn, and H.J. Ehrlich, Cold Spring Harbor Symphony Symposium, and on quantitative biology. Credits to all of them. So this is about PSA, which is the invention of Carrie Mullis, who has a US patent. Number 4683202 has had a tremendous impact on many research areas. So this research has had an impact in many areas, including molecular biotechnology. The capability of generating large amounts of DNA by amplification from segments of cloned or genomic DNA has facilitated the cloning of DNA versions of rare mRNA molecules, screening gene libraries, diagnostic tests for gene mutations, or physical mapping of chromosomes, and a myriad of other applications. In fact, the first study using PCR described a diagnostic test for sickle cell anemia, Psyche et al. In 1985. PCR was a unique idea that did not replace any existing technology. The power of the method is in its simplicity, sensitivity, and specificity. It utilizes a mechanism similar to that used by our cells to accurately replicate a DNA template. And it can detect and produce millions of copies from a single template molecule. In a few hours, and under appropriate conditions, it can be used to amplify a specific sequence in a complex mixture of DNA molecules, even when other similar sequences are present. Mullis received the Nobel Prize in Chemistry for his work on PCR, its polymerase chain reaction in 1993. Since 1986, more than 200,000 published studies have used PCR, one way or the other. Moreover, a Google search was with the phrase polymerase chain reaction yields more than 17 million hits. Its status as an indispensable method is well established, and considering the large number of PCR applications that have already been devised, there seems to be no end to its potential use. And now ATP sulfurylase to form ATP is the continuation of the former part. Now ATP driven Now briefly, the pyrophosphate generated by incorporation of a nucleotide is combined with adenosine 5' pyrophosphate by the enzyme, what we just read now, ATP sulfurylase to form ATP. In turn, ATP drives the conversion of luciferin to oxyluciferin by the enzyme luciferase, which generates light that is recorded by a photon detector. Before the next nucleotide is added to the mixture, ADP and any unincorporated deoxynucleotide triphosphate is degraded to its monophosphate form by the enzyme apyrase, which is an ADP diphosphorylase that removes the gamma phosphate from nucleoside triphosphates and the beta phosphate from nucleoside diphosphates. Because the natural nucleotide of DATP can participate in the luciferase reaction, deoxyadenosine alpha thiotriphosphate, DATP alpha S, which is used by DNA polymerase but not luciferase, is substituted for DATP in the reaction mixture. The amount of light generated after the addition of a particular nucleotide tends to be proportional to the number of nucleotides that are incorporated in the growing strand. The incorporation of any single nucleotide produces an amount of light that falls within a limited range that represents the incorporation of one nucleotide. That is, a 1-mer dinucleotide sequence, example AATT, CC, or GT, generates a more intense signal that falls within the 2-mer range. This more or less linear relationship holds only for stretches of about 8 identical nucleotides in a row. Homopolymer tracks. Generally, regardless of the DNA sequence in chemistry, homopolymer tracks are difficult to sequence accurately. During each incubation, the amount of the light signal is measured and recorded. At the end of the sequence running, the light emission are represented as a bar on a graph diagram, where the y-axis reveals whether the incorporation is equivalent to one or more nucleotide units, MERS, and the x-axis presents the sequence of the strand that is complementary to the template sequence. Sequencing using reversible chain terminators. For parasequencing, in which DNA is sequenced by synthesis, that is by doing nucleotide extension of a growing DNA strand, each of the 4 nucleotides must be added to the reaction sequentially in separate cycles. This process would be considerably faster if for each cycle all the nucleotides were added together. For this, it is necessary to ensure that the growing DNA strands are extended by only a single nucleotide during each cycle, and that the incorporated nucleotides are recognized individually. These objectives can be met with reversible chain terminates and 4-color fluorescence, which forms the basis of some new sequencing technologies. One approach entails capping the 3' carbon of the deoxyribose sugar with a chemical group that blocks subsequent addition of nucleotides, and attaching a different fluorophore to each nucleotide at positions that do not interfere with either base pairing or phosphodiester bond formation. Figure 4.31, DNA sequence determination of parasequencing. A, template strand blue background with primer sequence. Blue letters B, signal light intensities with 4 parasequencing cycles based on the template sequence in panel A. A parasequencing cycle consists of 4 rounds, where 1 round represents the addition of one of the 4 deoxynucleotides. Each round is followed by treatment with aqueous. A minus sign denotes no light emission, and the plus sign indicates the relative amount of light released after the introduction of a particular deoxynucleotide. Paragram based on the data in panel B and deduce nucleotide sequence of the strand that is complementary by the template sequence in panel A. This is the template strand. This is the table shown in the rounds, the deoxynucleotide signals, round deoxynucleotide signals. Pairs is added after each round. You have all the light intensity out here. The graph for that. Then you have the primer sequence going to the cleavage set and the linker. It goes with the blockage and the fluorescent dye. The primer sequence, the D primer sequence, and so on to the cleavage side. And after that, we can see that it's essential that both the 3' blocking group and the fluorescent dye be quickly and easily removed. Also, the decapping step must restore the hydroxyl group at the 3' position of the deoxyribose sugars to provide a site for phosphodiester bond formation with the next nucleotide. After fluorescent emissions are recorded, only 30 seconds is required to remove both the 3' blocking group and the fluorescent dye from each incorporated nucleotide, and to leave a hydroxyl group at the 3' position at the deoxyribose sugar. Cycles of nucleotide addition to the growing DNA strand by DNA polymerase, acquisition of fluorescence data, and chemical cleavage of the blocking and dye group are repeated to generate short sequence determinations reads of up to 36 nucleotides per run. Currently, read lengths are limited by difficulties in incorporating the fluorescence, nucleotides, and incomplete cleavage of the dye or blocking groups. The sequence by ligation is completed already. Now going on to the review questions. Question number 1. If your new DNA synthesizer has an average coupling efficiency of 98.5 degrees centigrade, what overall synthesis would you expect after synthesis of 50 more DNA hybridization probe? 2 is suggest 2 name strategies for synthesizing a 0.5 kb gene. Discuss the advantages and disadvantages of both methods. What is a linker? How is it used? What are the essential components of a PCR used to amplify a specific DNA sequence of DNA? Outline the steps of a PCR cycle. Discuss the significance of long-term templates and short templates and their prevalence as the number of cycles increases during PCR. Discuss how PCR is used to synthesize a gene. What is a primer? What are the key requirements of an effective primer? Describe some of the techniques that require primers. What is a dideoxynucleotide? What is a dideoxynucleotide? How is it used to determine the sequence of a DNA molecule? Why is it necessary to make DNA single strand? Determining its sequence. Draw the autoready derived from dideoxynucleotide sequencing CCTG as shown here. Outline the basic features of parasequencing. How are incorporated nucleotides recognized after each cycle sequencing by synthesis using reversible chain terminators? How does this differ from parasequencing? How are short sequences of a DNA fragment determined using oligonucleotides and DNA ligase? Describe the basic features of whole genome. Shotgun sequencing. And what are the advantages of cycle array sequencing compared to the shotgun cloning method for large-scale sequencing? Describe how 1 million or more copies of DNA molecule are attached to a single bead. This ends up the lecture on chapter 4. The lecture on chapter 5 will follow soon. This closes the lecture here. Thank you. Make notes and be prepared. Send in all your queries and any requirement for any clarifications to the comment section of this particular lecture. Thank you.